Well, good afternoon, everybody. We're going to hear about Alzheimer's from a neurologist perspective. So let's get started. So I'm going to start with a slightly historical perspective. These are slides that I wrote last night, and then I found a bunch of pictures to go along with these. So we'll talk about it, and then I'll show you pictures so we can go over it once. And then I'm going to show you PET scans, so we'll go over this a second time. So Alzheimer described his disease in 1907. And there's a big debate in the history of science as to whether major discoveries occur because someone has a great idea or because new technologies push the whole field forward. And I think for Alzheimer, it was absolutely a case of new technologies pushing the field forward. And so what happened at the turn of the 19th century into the 20th century is that better methods of looking at brains microscopically were developed, and people just picked a bunch of diseases and started looking at them, and they found all these interesting things. So when they looked at the 3 people with onset of their dementia before the age of 65, 2 56-year-olds and a 61-year-old, Alzheimer found 2 lesions that he described. One were neurofibrillary tangles, which occurred within the cell body of pyramidal cells, which is one of the common kinds of neurons in the brain, and extracellular deposits that he called plaques or senile plaques. Now, we skip, because I cannot be organized, to the next to the last bullet. What that says, until the late to mid 1960s, Alzheimer's disease was considered a rare cause of presenile dementia. So it was known, probably, certainly for those people who wrote the Bible and for other ancient literature, that many people, when they got to be quite old, had cognitive impairment. And until the 1960s, it was hypothesized, as someone went around to make hypothesis, that the cause of dementia in the people in their 70s, 80s, and 90s was a vascular disease. And Alzheimer's disease had been known for almost 60 years, but it was felt to be quite rare and of interest only to neurologists who like eponyms, that is, people's names. Then in the 1960s, in the UK, a group of Tomlinson was a neuropathologist, Roth is a psychiatrist, and I don't recall what Blessed was, but it might have been a neuropsychologist. They did very simple-minded studies, and I'm going to describe them because we repeated them. So if they were simple-minded initially, I guess they're extremely simple-minded when someone repeats them 20 years later, but, or actually it was about 10, 15 years later. But what they did was they took older people who they assumed would die within their funding period, and these are people in their 70s and 80s, and they gave them a brief mental status test. It wasn't the mini mental state, but for our purposes, we'll call it the mini mental state, and I switched the scores. So they gave these people the mini mental state exam, and then they waited till they died, and they looked at their brains. And what they found, to their surprise, was that half of the patients had the only pathology they could identify was Alzheimer's disease, which was supposed to have occurred only in younger people. And for another quarter, they had a mixture of vascular changes and Alzheimer's disease. So they could use the S word, or later people used the S word to describe these studies, that is S as in seminal, because what it did for the Alzheimer community was change this disease from being a rare disease of little public health consequence to a disease of enormous public health consequence, particularly because with advances in medicine and better food and other factors, people were living longer. And the older they lived, the more likely they were to become demented, because as you know, the prevalence of dementia increases from the 70s to the 90s. And in the 90s, it's about 50 or 60%. Okay, so I wanted to emphasize those last two bullets, so now I can backtrack a little bit. So the third bullet was that by the 1920s, it was recognized that the staining properties of the plaques were similar to other proteins that were called amyloid. So the first time I heard of amyloid in medical school was when they were discussing multiple myeloma. And I really haven't thought about it in the last 50 years, but what I do know about amyloid is that it refers to proteins that have a specific beta-pleated sheet configuration, and they have specific staining characteristics. There are many kinds of amyloid. Amyloids can be normal proteins, and there's one kind of amyloid that exists in Alzheimer's, which is quite important. Okay, so by the 1920s, it's recognized that the staining property of these plaques was similar to, was like amyloid. And also in the 1920s, though I'm pretty sure of this, I'm not 1,000% certain, that some of the cases of early onset Alzheimer's were hereditary. Okay, so that gets us up to the late 1960s. All right, so as I prepared this, I have at the end, what is the future? And we're going to talk about monoclonal antibody this and monoclonal antibody that and blood tests X. This as tomorrow's an election, and as I am a lifelong liberal Democrat, and the dean is going to call me up and tell me I'm not allowed to do these things. But I look at this as a measure of health disparities. And so this is a peek at the end of the lecture. So what can one do in 2020, and probably, which will also be true in 2025, and will also be true in 2030, is that one needs to practice lifelong wellness efforts. And what do I mean by that? Don't get hypertension, which means don't get real fat, which means keep on exercising, which means don't get diabetes, which means don't get real fat. Now, to do those things, one needs to have a fairly, this is a sweeping generalization, but it's always difficult to do those things. And if you worry about where you're going to live, or how you're going to feed yourself, or how you're going to get health care, or how you're going to pay for your children's education, that becomes even more difficult. So I think that our hopes of decreasing the dementia epidemic need to start, in my opinion, with these things that we know how to do, but we just have to have the political willpower to actually make happen. And then what else will happen in maybe 5 or 10 years is that people with genetically driven neurological diseases that present in middle age will be picked out in their 20s or maybe earlier, and they may have lifelong kinds of prophylaxis, and then disease-modifying efforts aimed at decreasing toxic proteins will be used. But that's the punchline at the end, and I just wanted to get my first liberal bullet point in. Okay, so what else did Blessed and Tomlinson and Roth teach us? That tangles in the neocortex, that's the outside part of the brain, to be distinguished in this discussion from the hippocampus, almost never occur in cognitively normal elderly. When neurofibrillary tangles are frequent in the neocortex, the people are always quite demented. Some tangles occur in the entorhinal cortex, the area CA1 of the hippocampus. Now, I'm sure you hate those words, so I'm not, I'm not testing on anything. But what you probably do need to know is that the hippocampus is in the medial temporal lobe, bilaterally in the front part of the medial temporal lobe, number one. Number two, that information from all over the brain converges down there. And number three, and this is probably most important, that we know because of the patient HM, which I'll describe in a minute, that's absolutely important for anterograde memory. That is, if you don't have a hippocampus, you cannot make new memories. So here we have the pathology of Alzheimer's disease affecting the entorhinal cortex and the hippocampus, and that's one needs to make memories, and the presenting symptom of Alzheimer's disease is GDOG. I can't remember what I did yesterday, but I can remember what I had for dinner 25 years ago. And that is probably much of the reason why that's the symptoms. So normal elderly have a few tangles there. Demented elderly have lots of tangles there. Okay, so here's a picture. Actually, and I believe this is true, although I didn't. So I went into Google and I typed in senile plaque, and this picture came up pretty quickly. And I believe actually I'm a co-author on this paper because the first author is Dennis Dixon, who was a neuropathologist we worked in, and I believe it's from 1968. And this is a double labeling, not 1968, 1988, a double labeling study. But what one sees are senile plaques and neurofibrillary tangles. So here are the senile plaques, and you can see they're big. I'll show you another picture where they put some marker in, and you can figure out that they're about 100 microns or more in size. And these are neurofibrillary tangles. And what basically these are the cell bodies, which are staining very darkly. And they're bigger. You'll see this in some other pictures. They look a little larger than the normal neurons. Now, the senile plaques have amyloid in them. I think this is a double stain. But they also have degenerating neurites. Now, neurites is the generic term for dendrites and axons. And if you think about it, if your axons and dendrites are broken down and stuck around a senile plaque, your brain isn't going to work very well because the information flows from the dendrites into the cell body, down the axons to another dendrite. And if those are all broken down, you're not going to work so well. So there are a lot of reasons to be cognitively impaired in Alzheimer's disease. Cells die, but even the cells that aren't dead, they can be problems with communication. Okay, so let me continue. All right, I found these other pictures, and so I want to cite the authors. And you can look up the paper if you want. Now, the point about this, and we can talk about it during the questions because I don't think I emphasize it too much more, is these are blood vessels. I guess these are arteries. I guess this is a vein and this is an artery. And they're using anti-amyloid staining, and what you can see is that there's a lot of amyloid in the wall of the blood vessel. And so part of Alzheimer's disease is amyloid angiopathy. The blood vessels get sick because of amyloid. And here we have another picture of senile plaque. And here you can see it is about 100 microns in size. And here are the neurofibrillary tangles. Here you can see the cells. And the cells with tangles look much bigger. And I think much of this, I think, is artifact. They may get bigger, but they don't get that much bigger. Okay, so for the last 20 years, there's been a huge focus on amyloid, on therapies against amyloid, and less focus on therapies against tau. Why that is, we can talk about. I'm a bitter old man. Before that, I was a bitter middle-aged man. I found this paper, and the reason I'm citing it is because I stole the pictures that follow. Okay, so here is their first picture in their review. And what you can see here is, here's the normal brain on this side, and here's the brain with atrophy. And Alzheimer's brains in general are smaller than age-matched people without Alzheimer's disease. Now, old brains shrink, just like old vertebrates shrink. And so a normal young male brain is 1,200 to 1,400 grams and probably goes down 100 or 200 grams over life. And Alzheimer's are still a little smaller, but not enormously smaller unless it's very severe. And which we may talk about is that specific areas of the brain tend to degenerate in a very specific order. So here they've shown us some pictures. Here is the neuron, here's the cell body, and here's the neurofibrillary tangle within this cell body. And here is the plaque coming out by the dendrites. But the essence of it is not within the cell, it's outside of the cell. And there's microglia cells proliferate and astrocytes proliferate. Okay? All right, so what are these tangles and plaques made out of? Well, the tangles are made out of tau protein. Now, tau is a microtubular associated protein, and I'm going to tell a personal story. I made the allusion to, or I didn't allude, I explicitly talked about silver stains is the reason for a major breakthrough. So I finished my neurology residency in 1980, and for the next 2 years, I worked in the lab at Albert Einstein with Peter Davies. He, in the late 1970s, was one of the first 3 investigators to report that choline acetyltransferase was decreased in the brains of Alzheimer patients, which was a big deal. And then when I was there, we started working on neuropeptides like somatostatin and substance P, but there was an MD PhD student in the lab who was playing with what was new technology at that time, namely monoclonal antibodies. And they had the simple-minded idea of taking some Alzheimer's disease brain, which they had lots of in the lab, grinding it up, putting some adjuvant on it, injecting it into a rat or a mouse, and raising antibodies, and then making monoclonal antibodies from those cells. And they had a monoclonal antibody that was exquisitely sensitive to Alzheimer's pathology. And then they asked, well, what was the size of the protein that this antibody was binding to? And they said, gee, it's 68,000 Daltons. And they knew that 68,000 Dalton happened to be the size of tau, but for reasons that I don't quite understand, they didn't really think it was tau and didn't make that claim until other people did after that. But at any rate, tau is important for stabilizing the microtubules. So here we have the axon. And what does an axon do? You know, an axon in the nervous system can be 1 meter long. And it's got to get neurotransmitter from the cell body all the way on here, and it's got to get junk on here back to this cell body to get destroyed, and so they can communicate with each other. And microtubules are key to move these things along. So if your microtubules are sick, you're not going to do that, and the cell is going to dysfunction. And normally the tau protein is on the axons, but in Alzheimer's disease, it gets misplaced, and you can see it in the cell body or at the cell body dendrite junction. Okay, moving along. Now I'm going to give you a picture of amyloid, and then we'll get to some other things. So what this is supposed to be, here's the APP gene. So this makes the protein called, this is amyloid precursor protein. And here's the protein that it makes. I guess it's like 750 amino acids long, and it's broken down into a bunch by these enzymes into little pieces, including the A beta monomer. Now, what do I want to say? Okay, I'm going to tell you one more story about APP, and then we'll move on. So, as you may know, people with Down syndrome who have 321 chromosomes always get the pathology of Alzheimer's disease in their 30s and 40s. And most people, I think, believe that on top of their already usually impaired cognitive status, they get even worse cognition. So they get demented on top of their baseline cognitive, intellectual disability. And it's known, of course, that people with Down syndrome have trisomy 21. So then in the late 1980s, early 1990s, efforts are made to try to isolate the amyloid precursor protein. And guess where they found it on? They found it on the 21st chromosome. So this was really cool. The notion was the reason why people with Down syndrome get Alzheimer's pathology is because they have 3 copies of amyloid precursor protein gene, not just 2. And let me continue. Okay, so here we have some more pictures. I think I covered this okay already. All right, now I have to, this is my, I don't think it's my last hurrah, maybe it was my first hurrah. So I talked to you about the studies of Blessed Tomlinson and Roth published in 65 and 68. So the man who was the chair at Einstein when I was a resident, Robert Katzman, had a number of grants to look at causes of dementia in the elderly, and they did exactly the same thing that Blessed Tomlinson and Roth did. The difference was, instead of doing it in London, they did it in the Bronx. They recruited cognitively intact people between the ages of 75 and 85, and they followed them, and they tried to talk them into giving autopsy consent, and some of them did, and some of them died. And when they died, we had the opportunity to look at their brains, and the pathologist looked at their brains, and I had decided whether they were demented or not. And working with pathologists, I just put the data together, and this paper came out of it. So you can see the title of the paper was that non-demented elderly subjects had Alzheimer's pathology, which was, this may not have been the first report, but it certainly was an early report. And so I couldn't get the paper because it was before PDFs, and at least it wasn't on PubMed, and I couldn't find it on my computer. So I sort of recreated it in the Excel data set. So here on this axis, I guess, is the mini mental state exam. So here's 30, which is, is normal, and here's 0, which is, is really bad. And here is cortical tangle count. And what we plotted and what Blessed Tomlinson and Roth plotted 25 years before that was that if you have a lot of tangles down here, your mental status score is pretty bad. And if you have very few, it's not so bad. And the correlation coefficient, I cheated here, but it's about 0.6, explaining that you're explaining about 36% of the variance. And here is the slide for plaques. I won't go into all the details, but the point here is that some of the people with plaques were quite demented, but some weren't demented at all. This was the basis of the paper. And we use the term non-demented because we couldn't say 1,000% that they were absolutely normal, but it was clear that they weren't demented. And here there's still a statistically significant correlation, but it's not as strong. And everybody has said since then, and probably before then, that taus account for cognitive impairment much better than tangles. So why do I make this point? Well, I make this point because armed with this information, the Alzheimer community then spent 20, 25 years searching amyloid as a treatment for Alzheimer's disease, which because of this and other things, I always was somewhat skeptical of, but I'll explain to you the rationale why amyloid, although I don't think I understand all the rationales. All right. So what could we say? I made these points. Common and non-demented elderly. And this will be, you'll see this when we look at the PET scans for amyloid, and they're not very specific, especially in old people. Whereas, unsurprisingly, PET scans that use tau are quite specific. All right. I talked about this, I talked about that. I don't have to go through this. And tau protein, I think we covered all of these points, so I don't have to go through those again. All right, so now we get to the genetic studies, and this is in the early 1990s. And so I had mentioned that for 70 years, it was known that some people, maybe half of the people who got Alzheimer's disease before the age of 65, had a first-degree family member who also had dementia, and I've had the occasion to treat some of those people and to deal with their children, and it's, that's what doctors do. They deal with some serious illnesses, some of which are genetic. And so in the early 1990s, 3 proteins in particular were found for which mutations in these proteins always were associated with Alzheimer's disease. And so these 3 proteins were APP, the gene that actually made the amyloid protein, plus 2 other genes that cut the precursor protein that we saw on the earlier slide. So I think this was the set of studies that pushed this Baptist Taoist controversy, so this is, people are so clever. So the people who liked beta amyloid were called Baptists, and the people who liked tau were called Taoists. And since the genetic studies never showed that mutations in tau were associated with Alzheimer's, whereas mutations in tau were associated with other much rarer degenerative diseases, such as some frontotemporal dementias and presenile and progressive supranuclear palsy. The thinking was, amyloid was driving Alzheimer's disease. I don't want to talk about that. All right, so now in the late 1990s, techniques developed to make transgenic animals. Now, the reason I made this slide about this no naturally occurring animal model of Alzheimer's is because if there were a naturally occurring model, one would use that to study Alzheimer's disease. And anyone who's owned dogs, our dogs died before they were too demented. But I think cognitive impairment does occur in elderly dogs, and they can get plaques in their brains, but not tangles. So we can have cognitively impaired older mammals that are not primates, and primates as well, get cognitively impaired. Why they get cognitively impaired is not entirely clear. We can talk about that at the end in some of my other work, which was talking about dementia in the oldest old. But so there's no naturally occurring models, so scientists decide to make a naturally occurring model. And they put human amyloid genes into mice, and they overexpress the amyloid, and the mice's brain gets some Alzheimer-like pathology, and they get some cognitive impairment. And so now there are multiple animal models of Alzheimer's disease. This brings us to the late 1990s. And then around the late 1990s, 2000 group, and I don't recall the names offhand, did what to me was a very counterintuitive experiment. Although if I had thought about ALZ 50 harder, that antibody that the MD PhD student in our lab made, I might have come to the same idea. What these people said was, all right, we have an Alzheimerized mouse. Let's try to get rid of the amyloid that we put into it with antibodies against amyloid. So they took some, just as the medical student in our lab, his name is Ben Wells, and he has a name, did. They made some antibodies against Alzheimer brains and I guess against amyloid, and they wanted to see whether treating their transgenic animals with immunizing them, so that they actively immunizing them, so they raised antibodies against amyloid, would get rid of the amyloid in their brains. And they said that it did, and it apparently did. And they looked at the brains, and there wasn't very much amyloid in the brains, and it's not clear whether the mice got any smarter, but clearly they didn't have amyloid in their brains. And pretty much based on these studies, clinical trials were started. So we're now in the early 2000s. And so the question that I never quite understood, maybe Michael can help me with this, with your ideas, but I think in part it was because amyloid was easier for the labs to deal with. Part they were impressed with the genetic data, partly because they did this first and it seemed like a good idea, partly because if amyloid came first, then maybe that's where you should start. There were all these efforts, billions and billions of dollars, and years and years and years of anti-amyloid clinical trials, and they used active immunization, passive immunization, enzyme treatment that tried to either decrease the production of amyloid or increase the destruction of amyloid. And by and large, until I guess it was May 2020, all these studies were thought to be negative. But I'll get to that in a minute. Okay. And that was my. Okay. All right, so this brings us to aducanumab. And I learned this Sunday night, that it was developed by a Swiss lab that was then licensed to Biogen. So I may have mentioned that my son is chief medical officer for a biotech company that recently went on the stock exchange. So he discussed with me the licensing agreements they have with their products. Other than that, I can't, he discussed what he's allowed to discuss with me. But I guess this is the way pharma works. They licensed this compound to Biogen. And the compound is, there were studies that I haven't read that some older non-demented people have antibodies, naturally occurring antibodies in their sera that reacts with amyloid. So the hypothesis was developed, and I don't know the status of this hypothesis, is that these people with antibodies didn't get Alzheimer's disease because they naturally destroy whatever amyloid is made in their brains. So the Swiss company decided to say, well, let's make antibodies like that. So they sequenced the antibody from the patients, and they developed monoclonal antibodies similar to the ones from the patients. And they licensed it to Biogen, and they said, it works. And I didn't spend a lot of time on the amyloid slide, but the amyloid metabolism is fairly complex, and you can go from the monomer form, which is cellular, to oligomers, which are a few of them, and then to big polymers, which are a bunch of the oligomers bound together. And one antibody will bind to one form and one will bind to another. And then the question is, which of these is the one that you should use? So anyway, that's how this works. And so I copied this directly, and I guess I was off by a year. And I'm going to just read it out loud. So this is like a Seder. On March 21st, 2019, Biogen and Eisai announced they would terminate all currently ongoing aducanumab trials following interim analysis that predicted that their longitudinal studies with their very nice acronyms would miss their primary endpoints. And in April 24th, 2019, Biogen announced it would not initiate an anticipated phase 3 secondary prevention program for this drug. Okay, next bullet. On October 22nd, 2019, Biogen announced that the interim futility analysis was wrong, and that subsequent analysis of a larger data set instead showed that it did meet its primary endpoint. And I believe this was only for a subset analysis for people on the highest dose of the drug. And it met the primary endpoint, which is the CDR SB, which stands for the Clinical Dementia Rating Sum of the Boxes, which is basically an ADL scale that asks the caregivers what the person could do and what can't do. So it's actually a sensitive and not that noisy a measure. And they declined on secondary endpoints such as the MMSE, which is impressive since it's such a damn insensitive test, and these other measures. Okay, so that's the first slide I have about this. I have wondered whether the people who did the initial analysis or decided to announce it were fired. I don't know the answer to that. Maybe they were given a raise. What acetylcholine actually does, as it's the drug that we still use all the time, I'm going to put aside perhaps until questions. Okay, and I wanted to make these points, but okay. So based on these data, Biogen and their collaborators applied to the FDA, and a decision is pending in early 2021. And so we shall see. I think that this tau therapies are more promising, but we'll get to that. All right. All right, so what I'm going to talk about in the next 10 or so minutes are Alzheimer biomarkers, Alzheimer PET scans, and I'm not going to present any data on anti-tau therapies, except that they are ongoing, focused on various parts of the tau protein. To my knowledge, none of those have been, I think far fewer have been, clinical trials have been reported, and I'm not aware of negative ones that are reported. Michael probably is. At any rate, so what are the biomarkers? Well, it's been known for around 20 years that you can look in cerebrospinal fluid, and people with Alzheimer's, their amyloid level tends to be low. The hypothesis is it's all stuck in the brain, and their phosphorylated tau level is high. What I learned today, 2 hours ago, is that as the Alzheimer's disease advances, the phosphorylated tau levels actually drop. But nonetheless, it's a very useful biologic marker, except people don't like to do LPs. And then in 2020, May 2020, it's reported in Lancet Neurology that phosphorylated tau in the blood could be a useful marker for Alzheimer's disease. And this was done with 4 different prospective. They were retrospectively used 4 different data sets that had been collected prospectively. So these data, the samples were stuck in a freezer. The investigators said to the people who controlled those samples, can we use them? They said yes. And so they were able to get the data without doing another 30 years of research. And we're also going to talk about deoxyglucose PET scan, amyloid PET scan, and tau PET scan. So and I'll show you some pictures of those. Okay, this is not in order, so I'm not going to talk about that. Okay, so here are the pictures. So first we have, this is the paper by Bill Jagist, and this is for the Pittsburgh compound. I'm going to go forward and start with the glucose hypermetabolism, then go back to the amyloid PET scans, and then end up with the tau PET scans. So the deoxyglucose PET scans have been available for 20 years. We can order them ourselves. I have ordered them several times, maybe 20 or so times in my life. And I tend to order them on people who are young, that is 40s, 50s, 30s, early 60s, for whom the clinical diagnosis is unclear, many of whom I think have frontotemporal dementia. If I think that a young person has autosomal dominant Alzheimer's, I try to do the genetic testing, and if it shows the mutation, then I can quit. I don't have to do any more studies, and I've gotten the genetic information that the family may or may not want to know. But obviously, the family has to be counseled before we go in that route. So what does the deoxyglucose scan do? You inject glucose, and where it's in areas of the brain that don't work as well, you don't burn it up as fast as areas that work just fine. Now, understand, these are resting scans. These are not activated scans. People are in a scanner and they're told just to relax, not to think not to think lovely thoughts. One point I would make is I have ordered all my PET scans from one place, North Shore. I strongly discourage using downstate, which I don't think do brain PET scans, or any other place. North Shore has done thousands and thousands of them. They've done much research on it, and I've gotten some PET scans that have been obtained by others in places, in private imaging places, and I do not recommend it. I think the data are pretty crummy. And what one can see here is, I guess here on the top row, these are the, let me see if I can come over here. So this is left lateral brain, left medial brain, right lateral brain, right medial brain, and this is inferior brain. And so here in the medial brain, you can see the hippocampus and entorhinal cortex, the medial temporal lobe area that we're talking about. Here we see the inferior temporal cortex and we see the angular gyrus. Now, all of these areas are going to correlate with symptoms of Alzheimer's disease. This is the anterograde memory problems. This is naming problems. These are what are called areas of polymodal association cortex. So this is where the brain integrates areas from like auditory information with visual information. And these are the areas which tend to be impaired early in Alzheimer's disease. Now, I never get them if I think someone has Alzheimer's because, well, we can talk about it, but the issue is how is it going to change your therapy? And a point that I made last week, I believe, and I'll make again now, is that when DMTs, disease-modifying therapies, are available, and if the disease-modifying therapies have side effects, since they almost certainly will, then the onus will be to get as much information as possible to be certain that this A is Alzheimer's disease and not vascular dementia or Lewy bodies or Parkinson's disease, and B, also to probably monitor the course of the illness. But until that time comes, all this information is really not going to influence any clinical decisions. So in my opinion, most of the time, it's not indicated. It's neuroradiologists with the right algorithms can quantify the amount of cortical thinning. And all this is saying is that the areas that on deoxyglucose scan don't show a lot of glucose metabolism are also thin. So now let's go back to the amyloid scans and then we'll go to the tau scans. Okay. So what we see here is lots and lots of amyloid in many regions of the brain. And I think this is somebody with somewhat advanced Alzheimer's, but you can see the temporal lobe is totally coated with amyloid. Frontal lobe is virtually coated as well. Medial parts of the brain, the cingulate gyrus is done. What is this called? Blocking for a second. The precuneus is covered, occipital lobe is covered, so you have amyloid all over the place. And one of the concerns that we knew in 1988 is that the relationship between amyloid deposition and disease progression is not so clear at all, or there is not a good relationship between amyloid deposition and disease progression, number one. Number two, our studies and many, many other studies said, gee, you can have amyloid in your brain and not be cognitively impaired. So if you take an 80-year-old and he's got amyloid in his brain, to conclude that 5 years from then, he's going to be significantly demented from Alzheimer's, probably is a mistake. That's the problem with amyloid scans. Now, if the person's 50 or 60, I think it's a different story. But when the older the people are, the less specific it is. Let me continue. Okay, and here they wanted to show you that there's an overlap with the default network, which I'm not going to go into. And here's someone with mild Alzheimer's, and you can see that they already had a fair amount of disease in this frontal region. And at this time, there'd be absolutely no tau there. So why one gets amyloid there and tau in a different place has never been explained as far as I can tell. All right, now we'll get to the how, where the story is different. Okay, so what we have here is now we're doing PET scans with the compound that binds tau or phosphorylated tau. And so the Brocks were Austrian neuropathologists who did something else that was simple-minded. I guess they like simple-minded things. And the Brocks had about 1,000 autopsies on Alzheimer's disease patients. And so I presume they paid a grad student or a postdoc to say, take these 1,000 people, and what I want you to do is I want you to rank them in order from the least pathology to the most pathology. And they wanted to see whether there was a consistent pattern as the pathology got worse and worse. And they found, gee, that there was a consistent pattern. And so they took these 1,000 brains and divided them into 6 groups. And groups 1 and 2 had very little tau pathology, and groups 5 and 6 had a great deal of tau pathology, and 3 and 4 were in the middle. And the groups 1 and 2 only had temporal lobe pathology. And if you do a tau PET scan on somebody with mild Alzheimer's, this is all you'll see. Medial temporal lobe, which we kept on talking about over and over again, showing. And what these are doing is they're showing the activity to make it dramatic. They're subtracting people with stage 1 and 2 from the controls. So it makes it stand out. Okay. Now, when we go to more moderate disease, you can see by this time the inferior temporal lobe is really smacked. This is where the worst pathology, and now inferior frontal lobe, medial frontal gyrus is starting to get hit. And again, it's not surprising that problems with language are quite dramatic at this time. Problems with praxis, which we rarely test, would be predicted from pathology in this region of the brain. So again, it correlates very nicely. And finally, this is stage 5 and 6, and you got this stuff all over the damn place. All right, and there's a paper from that. So now I'm running out of time, so I'm going to talk about the future again. So I'm going to repeat the slide that I said before, because for 15 or 20 years, I've been saying to people with, or maybe 30 years, for people with mild, moderate, and severe dementia, what they can do. And I say, stay healthy, do physical exercise, which is stay mentally active, don't get depressed. If you get depressed, try to stop getting depressed. But the issue is all of these things are almost impossible to do in this country and in most countries of the world unless you have food on the table, a roof over your head, some kind of meaningful work, and healthcare. So without those things, I don't think we're ever going to be able to deal with the dementia epidemic. And then we're going to start to pick out the people probably from history and blood tests who have middle onset of neurologic disease, such as early onset Alzheimer's. And for everybody else, what I suspect will happen is that when you're 50, your doctor will say, we're going to schedule a colonoscopy and we'll get you a blood test for phosphorylated tau. Maybe they'll have started taking it when you're 20 and get it every 10 years. And if they see a peak in your phosphorylated tau, they'll say, you need to go to the neurologist. So the neurologist will do his or her thing and probably get a bunch of studies trying to figure out whether this marker of phosphorylated tau is associated with any other evidence that you are likely to develop Alzheimer's disease. And so the blood test will cost whatever the blood test costs, but all these other tests will be expensive and take some time. But I suspect that if the person is positive on these tests, then we'll say we have some antibody therapy to give you, either vaccination or passive immunization, and maybe it will be against tau, maybe it will be against tau and against amyloid, maybe it will be, there'll be one monoclonal against one part of tau and another monoclonal against another part of tau, and one monoclonal against one part of amyloid and another part against another part of amyloid. And they might as well throw Lewy bodies into the mix, because if you, because that will come too, I hope. And then I think, as I said last time, I think there will be efforts to maximize brain metabolism and blood flow, because you got to get the glucose and the oxygen up there, and you will lose some neurons with aging, and they will probably, their mitochondria will be less functional. So if you can budge those up a little, it will be useful. And finally, I think there will be some efforts to tune and detune oscillatory networks. And with that, I will stop and thank you, and I will take your questions.